Okay, so, well, thank you very much for the invitation to present our work at this series of seminars. And I will try to be brief so that because we have a lot of material to present. So just to put these things in an historical context, the work we do is for applying deep learning and machine learning techniques to microscopy. And this fits into an historical trend towards quantifying microscopy with quantitative tools. So if you go back, this goes back even 100 years to the first studies by Perrin, who basically tracked manually, as you can see here, the trajectory of Brownian particles, and then used these trajectories to quantify their diffusion, well, actually to observe and measure the Brownian motion, and by doing that, to actually measure the size of atoms and molecules and convince the international scientific community that atoms exist. One striking fact is that before that, scientists still thought atoms were mostly a nice mathematical tool to understand reality, but they didn't believe they actually existed. Later, other quantitative methods were helped by the introduction of microscopy techniques combined with electronics, which permitted the more systematic acquisition of data than what could be done manually. And this came about around the 1950s, and has led to many techniques that nowadays are very widely diffused, such as, for example, techniques related with the flow cytometry, which is used in basically any blood analysis. And more recently, there has been this trend towards the use of tools from AI to analyze images obtained from microscopes. This tool include, for example, the invention of the UNet around 10 years ago, as well as a lot of tools for particle tracking and for microscopy image analysis that we have been also developed within my group and Carlos' group. And this leads to the advent of deep track and our technique, which has been what we have been doing the last 5 years, which was the focus of what we will present now. Now I pass the word to Carlo.
Yeah, so as Johnny was saying, the application of AI to microscopy is relatively recent, and the unit was applied for cell segmentation about 10 years ago. But since then, the progress has moved on quite fast. We've seen a lot of progresses, in particular in specific fields related to images. So cell segmentation is one, but another one is tracking. If you can move to the next slide, where nowadays there are a lot of tools also for following objects that can be cells or single molecules or vesicles, or also beads in colloidal materials. And besides that, other efforts, for example, still related to the field of segmentation include, for example, the cell pose, which moves from semantic segmentation to instant segmentation, and also other techniques that do different tasks, different application. For example, image mode transfer or virtual staining. If you can move on, in which basically we can move from an image modality like bright field or phase contrast to another one that allows us to specifically label cells region, like nucleus, organelles, and so on, without chemically staining the image, but just through the application of AI. I mean, all these applications provide a lot of tools. There are full zoology of tools that are more or less user-friendly and are available. If we restrict ourselves to optical microscopy, we can move to the next slide. So optical microscopy scans several scales, both from the spatial and temporal point of view. And if we only think about the cellular level, I mean, we are limited in the region between 10 and 100 microns and second to minutes. And with optical microscopy, if we move to the next animation, we can see that we can collect movies like the one shown here, in which the cells move. and move in a very diverse way. They often split, they change shape, and this movement is highly heterogeneous and highly dynamic. On top of that, I mean, if we don't limit ourselves to optical microscopy, to simple optical microscopy, but we include other technique like electron microscopy, localization microscopy, and so on, or fluorescence microscopy, we expand even further this range of spatial and temporal scale. And if we want to apply AI to these methods to improve their quantification, as Giovanni was saying before, we have several limitations. But the first one is that, first of all, most of the methods that have been applied so far to improve the quantification of images and movies are supervised. So in order to be able to use them efficiently, we need a lot of label data. This data, the quantity of data is basically justified by the fact that the stochasticity and heterogeneity of the data needs to imposes that we train our machine, our methods in a very diverse situations in order to learn as much as possible about the movement. And we have dedicated a major effort in these directions, specifically trying to reduce the amount of data required for training, and eventually obtaining a supervised method that we can just use our experimental data and get us information without any specific labeling or annotation. The common point of the approaches that we will describe today is the use of inductive biases. I mean, if we think about deep learning, can you move on? Yes. If we think about deep learning, fundamentally, I mean, we're applying statistics, we're doing optimizations. And the main difference from common from statistics is that we do this optimization using a lot of parameters, and that's why we need a lot of data. In order to limit the number of possible solutions, so in order to be able to do this optimization with less data, we need to apply some constraint. And we can apply constraints inherently, for example, designing smart architectures or using the appropriate data representation in our problem. One possibility is to leverage relations between data points. For example, when we apply this neural network, we inherently assume that our data are independent. So there are no relations, and that's why we mix them all together. But for example, when we use images, we use convolutional neural network because we want to enforce the locality of the image process itself. And similar, if you have sequences, we use networks like recurrent neural network that already include the sequentiality. In the cases where there are structure and relations in our data, but those are not regular or structured, like for example, in the pixel of an image, we can apply a different architecture that are based on graphs. And we will show that a few samples in which we apply graphs to different problems. The first two examples come from the field of object tracking, applied to where the objects in our case are cells or single molecules. And by tracking, we mean that from movies like the one that you are seeing now in the slide, we would like to determine trajectories. So basically, I understand where each cell goes at each step, time steps, in order to estimate parameters that are relevant from the biological point of view, and that can enable us to measure, for example, the effect of a drug or a treatment or a disease. And this process of tracking is typically approaching two steps. In this next slide, we will show the first, in which basically we analyze each frame, so each still image that composes the movie, and we want to detect the objects. Like in the example before, we have cells. We want to see in each frame of our movie where are the cells, so we detect where they are and get their position. And this is already an approach similar to the one that we described before, that, Giovanni, can you move on, please? That allows us to do this detection and localization, and can be approached in several possible ways, based both on classical methods of image analysis or on deep learning. And in this sense, we developed a method that is called Lodestar, and I guess Johan is going to explain you the principles from the next slide on.
Yes, so just to give an example of these methods that are, well, obviously you can use standard methods that will require thresholding and other techniques. This is an example of a very data and computation efficient method that we developed recently, and is based essentially on taking a single frame. So you take a single image, a crop of the particle that you want to track, and with that frame alone, you can basically train a neural network to follow the particle. And here you see it on several simulated examples data sets. You see that it's following the particle perfectly. The nice thing again is that you use a single crop for training the neural network, and you don't even need to know the position of the particle in that crop. And of course, then you can apply it to actual videos taken from experimental data. So here you see a few examples from the cell tracking challenge videos in which this method permits to actually track essentially perfectly the position of these various kinds of cells, being trained on a single cell, a single crop of a cell, without knowing the ground truth. Of course, the question is, how can you train a neural network on a single crop of a cell without knowing the ground truth? Well, I'm not going to go into the details, but the basic idea is the following. If I give you a picture of a cell and I don't tell you the position of the cell, you don't know where is the cell, obviously. But if I take the same picture and I translate it by a certain amount, what you know for sure is that the position of the cell has been translated by the same amount. So you can use this information to create an error signal that you can use then to apply backpropagation. This technique is essentially based on a mathematical principle called equivariance, and can be applied to all problems, not only cell tracking, that show this kind of equivariance in some way or form. So it's an example of this extremely data and computation efficiency tracking of cells in this case. So this is one example. And I guess now it goes back to Carlo.
Yeah, because I mean, now that let's go back to tracking. Now we want to reconstruct the trajectories. And for example, if we apply Lodestar, for example, to get the particle localization at each frame, we still don't know how to connect these dots. Basically, we want to be able to link the position of a cells with the same cells in the future and in the past. And this, of course, it's easy if we have a very sparse labeling, very sparse experiment. But when things are dense, and for example, in cases where the cells can change shape, it becomes more difficult. And ideally, you would like to use an approach that is able to understand all this variability and all this heterogeneity in order to provide the best possible tracking. And this is when we started to develop MAGIC, which stands for Motion Analysis Through Graph-Based Inductive Knowledge, which is a geometric deep learning approach based on graphs to do this linking to step. So basically what we have after the detection and localization, we end up with a series of localization, a series of coordinates. Now it corresponds to cells. And for each of these coordinates, we not only know the position, but we also know other information about the cells. We can calculate intensity, shapes, morphology, other parameters. Now, the thing is that besides the information about each object, we also know that there are relations among them. For example, we know the distance, or we know both in space and in time. We also know how frames it's distant and localization of banana. And basically, this construction inherently help us to build a graph. So a mathematical structure that associates to each node, so to each of these localization, the feature that I was mentioning before that tells us about the morphology and position. But we can also include information about the edges, like I was saying before. Can you click on the slide? Like, for example, the distance, in this case, for example, only the Euclidean distance between adjacent points. So we end up with a structure of this type, on top of which we can apply a neural network. The neural network, in this case, basically works at the graph level and updates node information based on the neighboring nodes. So it basically does a convolution, but as a convolution using a variable structure that depends on the proximity of each node and on the edges that connect this node with the neighbors. We can train these as, stay on the previous one, please. We can train these as, for example, in several different ways, but basically as a graph-to-graph network in which the network learns to do an edge classification. So we have an initial redundant number of edges that we have used to connect our dots, and we want to classify the edges in a binary way, those that do connect cells, the same cell at different times, and those that do not. In this way, we can basically start from the redundant graph description that has many edges and reduce it only to the final trajectories, to the connection of the same cells at different times. And this works very, very well, as in the example that we will see now. Basically, this is a movie from Benchmark, from the cell linking challenge. Same color here means the same cells at different times. And you can see that the method works very well. We have a very super high accuracy in spite of the fact that the shape of the cells is continuously changing and the cells are also dividing. The important thing here is that the slides are moving back and forth continuously. I don't know if it's me.
No, it's me.
Okay. So basically, the important thing here is that we trained Magic on only one movie, and still we get extremely good performance that are competitive with the performance of the top scoring teams in this benchmark. And even if we move on to the next one, even if we change the temporal and spatial scale of the movie, we still get very, very nice tracking with super high performances. And we also observe that works very well for transfer learning. So basically, you can train on one movie and apply on a movie of different cells with a different background, different scenarios, and so on, and still is able to get a very, very high performance. Can we move on? Okay, so far we have been discussing about things that move, so movies in which we apply microscopy to determine dynamics. But graph neural network and inductive biases can also be applied to other types of analysis. For example, the clustering of localization microscopy images, which is what we did with Miro, which is another method based on geometric deep learning for spatial clustering of single molecule localizations. I don't know if you're familiar with super resolution microscopy. Basically, let's first think about a conventional image done with fluorescence of a very dense object. For example, in this case, we observed a nucleus of a mammalian cell in which we labeled some histone protein. The histones are very dense because the chromatin is very compacted. So if we label each single molecule and we image it because of with fluorescence, because of diffraction, we see a very, very blurred image. So we do not have details about the molecular organizations. But this is just because of the diffraction and of the density of these proteins. If instead we are able to switch most of them off at some time and then stochastically activate them in such a way that at each frame we only have a few molecules that are on, by collecting information on the position of the molecules that we can clearly distinguish, we are able to resolve much better the image. And what we get is that if we move from this blurred image to a very much detailed image in which we start to see structures. If we can click. So basically here we start seeing these structures that are formed by these proteins inside the nucleus of the cell, because we have improved the resolution using this trick. But in most of these cases, we at the end of the day want to do something quantitative. So we want to get numbers out of this spatial organization. And typically, we want to do that from the output of this imaging process, which is a point cloud. Can we move to the next slide? Again and again. So basically, imagine that we have some structure in our data. The process, the stochastic process of associated to the image, corrupted with some stochasticity, some noise. So we still see some sort of structure, but this structure is more noisy. But we still would like to identify structures and quantify them. So the first step in this case is a sort of segmentation. We want to be able to detect which of the localization that we see in this point cloud belongs to a specific structure, in this case, a specific ring. And again, we can use this information to build a graph. And also in this case, we have information about the points. This is the final result, what we want to get. No, basically, we want to be able to get to color each localization as part of the underlying cluster. And in order to do that, we can build a graph. And for example, use a neural network, a graph neural network that does the following. It basically takes the information, spatial and relational, from the graph, and in a supervised way shrinks the point that belongs to the same cluster, in trying to collapse them toward the center of the cluster. And basically, by learning this, the output of the network is a vector field that leaves the noise points alone, but squeezes those that belong to the same cluster together. And this can be done, for example, applying this simple block recurrently. And at the end, the network learns to identify the cluster, and basically it shrinks all the localization that form the same ring toward a circular spot, toward the same region. And once you have that, basically you can use whatever clustering algorithm like dbScan, because now it's an easy problem. You have spots that are very well isolated and are circular to get your segmentation, to get your classification. You can move on. So we quantified that using data from the simulation from a benchmark that was done for these tasks. Again, we use different shapes, different densities, several metrics to understand the performance of our methods, and we basically we do much better than all the other existing methods in all the cases. In particular, the performance of these approaches are very relevant when the shape is not trivial. When we have shapes that are either elongated or non-convex, like for the cases of ring. But in real life, I mean, typically you have structures that have a hierarchical structure, a hierarchical organization, because you can have these rings, but each ring can be formed by spots that might have some symmetry. In this case, it's a eightfold symmetry. And in the same process, you would like to be able to cluster the rings, but also cluster the spot that form the ring. And with our architecture, we can easily do that, because basically on top of the training that I already described before, we can put an intermediate step that learns about the first level of clustering, one of the spots. And if you click, you will see the result of that you can also cluster using another algorithm and whatever algorithm. And then, so basically in one training, you get both information about the rings and information about the structure that compose the ring. Also, in this case, we have benchmarked on simulations and also on real data. Those are data from the group of Jonas Rees. Those are nuclear core complexes, so the structure that form in the nuclei of cells and have this eightfold symmetry that I was mentioning before. And using this method, we are able to recover information that is very similar from the quantitative point of view to the one that they recovered in the original paper, but they did that using a very specific and dedicated algorithm. In our case, we use, an approach that is general and can be applied to any similar process. And we get a very good result. On top of that, I mean, so far I described the problem, which is a multi-scale. Basically, you have a hierarchical cluster, and you can simultaneously cluster these two structures. But we can also apply it to simultaneous clustering and classification. So basically, let's suppose that in an image you have two different shapes. In this case, we use rings, and with C-shape. The algorithm is able to learn the difference between these two and simultaneously classify the structures. And notice that this case is not trivial, because basically two C-shapes that are one facing the other form a ring. So the algorithm really needs to learn the detail of the structure in order to be able to separate these two different shapes. And then we go to the, so basically so far the methods that I described are a few shots, in the sense that you can train them using a very low number of examples or annotated data. In the case of mirror, the clustering, we have been able to train it to very good performances from one cluster alone and generalize it to many others. But as I was saying at the beginning, the final objective was to get something that is completely unsupervised. And this is what we try to do with Gaudi, which is another method that I'm going to describe briefly. In Gaudi, again, we use a graph autoencoder, and the problem that we want to face is slightly different, it's more general, although it has application in imaging as well. Can you move, Johnny? So basically, consider that you have a system. Typically, we talk about complex systems that you are able to describe as a graph. This system is characterized by physical parameters, and you want to get information about this system in a latent space in such a way that, if you have two similar graphs describing similar systems, so basically, if the physical parameter describe two systems are close in the parameter space, the representation that you get in the latent space is still close. Why we want to do that? To generate some continuity, because suppose, for example, that you have a system that is evolving in time, you want to be able to evolve the system also in the latent space without moving in a region of the latent space that is very far away. So you want to recreate and maintain the continuity and the structure of the physical manifold into the latent space. And the approach for doing that is basically, as I was saying, an autoencoder, in which we simultaneously shrink the information, which basically we downsample and then upsamples our graph. We pass through skip connections the semantic information about the connection of the graph, but what we reduce is the information about the nodes, so the nodes features. And in this way, we shrink progressively to the bottleneck when we get this latent space. And this latent space needs to capture the physical information in keeping the structure and the continuity that I was mentioning before. We applied it to many different cases. I will just show an example from imaging, since we are discussing imaging in details. And this is, again, those images are rendered images from a localization microscopy, so the super resolution technique that I was mentioning before. In this case, instead of showing the point clouds, I'll show a rendering with some Gaussian convolution that produces these images. And basically, those correspond to different structures. One on the left, you have a ring, and on the right, you have a spot. And because of the noise and other artifacts of this imaging process, we have a stochasticity. So the same ring structure in other cases in this case, you clearly see a ring, or you see something that looks like a ring, and you can distinguish from the spot that it's on the right. But in other cases, like the one that are going to appear as soon as we click, although the underlying structure is a ring or a spot, you cannot clearly define it because of this stochasticity associated with the imaging process. But if the information is there in the localization and the autoencoder, the graph autoencoder is working properly, we still must be able to get this information in the latent space. And this is what is shown here. Basically, we get a 8-dimensional latent space, and then we do some dimensionality reduction using the principal component analysis. And we see that we can get a quite clear separation between structures that is difficult to tell apart by eye. So the architecture is really learning the underlying structure of the data in such a way that it's able to structure the latent manifold in a sensible way. Just to give another example, those are data from CAN dataset. So basically, those are data from magnetic resonance imaging that basically are data from human patients that tell about the brain connections of these patients. And we also process them to the using this architecture. And what we see in this case is that the architecture is learning information about the age of the patient, because in the same representation similar representation as one we see before in the next slide, we see that it's structuring the patient, the elder patient from the young one in different clusters, in different region of response. So it's capturing dependencies in the brain graph that can be associated to the ages of the patients. And I guess with this is basically what I
Professor Carroll.
Yes.
I wanted to understand a few things about the previous slide where Gaudi is being applied to brain scan images, I think.
Yes.
And my intuition and why I'm wondering, maybe I missed this about the data input of the graph, and also looking at it, then the output from the graph going into the graph autoencoder. And now I'm trying to understand the input data structure and the output at the end. And I had not envisioned this as the image scan. Now, my question is, what exactly are we capturing from the brain image scan, and what works as the data input to the graph, but also maybe an insight?
The image scan is processed in such a way that for each region of the brain, which is a node, you get some features. And basically the information, this is what we use to build the graph. And then the autoencoder just tries to reproduce the same graphs. The information about the edges is passed at each level of the upsampling and downsampling directly. What is progressively reduced is the information about the nodes.
Okay, so is this like modeling like brain activity, like the entire brain, and looking at every node within the brain?
Yes, this is not every node, but I mean, it's looking at nodes within the brain, and basically, I mean, I don't know if you see the metrics that are represented in the bottom part. So basically, you have a set of features that describes the type of connection between nodes. Maybe Giovanni can have more information about this part, because
I mean, the graph that this CAN dataset is a standard dataset, which includes both MRI and DTI data. DTI are diffusion tensor imaging data, which basically image the white matter tracts in the brain, which is what we use then to calculate what is the connectivity matrix for each subject. So we get a graph from that. And then, as Carlo was saying, for each node, which is a brain region, corresponds to one brain region, we extract from MRI data some measures like cortical thickness, for example, just how many neurons there are in that region, how thick is the cortical region in that region. And then this produces a graph that can be analyzed by Gaudi.
Okay, makes sense. The last part is, and I think I missed this, but the structure of the data input into Gaudi together, like what can be thought about it? Yes.
It's always a graph. here we don't pass images, we pass a graph.
Okay, okay.
Also in
Okay.
this case, this is not passed as an image, but it's passed as a graph. So this is transformed into a graph first. So this is a set of localization which produces a graph very similar to the one you have here.
It's exactly like in this case, because it's the same imaging.
So here, imagine that this one, each of these points is a localization. Then we can construct a graph of the localizations.
Okay. Prof, and this is just to really understand the work deeply. In my thinking, I was at first, I was thinking like the entire structure of Gaudi is a graph, part of it, but also embedded with, like conjoined with the encoder, and that was my understanding first. So is that a wrong understanding, that the input to the encoder, decoder is just a graph as an input to the encoder?
The input is just a graph, yes.
Okay, but it's not like part of the entire Gaud architecture.
I mean,
I'm not understanding.
I mean, I think I understand this, is that graph neural networks are not graphs.
Okay.
I think that's the issue.
Oh,
A
okay.
graph
Okay.
neural network is not a graph itself. It's a neural network, typically a very simple one, like a dense layer, a fully connected neural network that operates on graph, but it's not itself a graph.
Yes, yes. Okay, I now totally understand what is going on. I get it. Thank you.
Maybe, okay, well, maybe later we can show the slide for that.
Okay.
But that's a very common point that might not be obvious from the name.
Yeah, this basically was just to try to wrap up the general message of the presentation. So there are many possibilities to design an AI system, because I mean there is a lot of space for improvement, in particular in microscopy and imaging. Inductive biases in general way, so like, you know, designing the optimal architecture, representing the data in the most proper way, or even choosing the right loss function can improve the performance, and you know, by creating these constraints that I was mentioning before. And there is, I mean, the thing is that there is no formula, there's no law to do that, to apply, no protocol to apply for doing that. So basically you need to apply your creativity and figure out what is the best way based on previous knowledge and the understanding of the problem. And then I guess, Joe, we have a few slides that Giovanni can present now.
Yes, very briefly, just to wrap up, if all these tools that we have been developing over the past 6-7 years have been developed within some Python frameworks. One of them is DeepTrack, which is a Python package for helping microscopy use machine learning. And this is available in the GitHub DeepTrack AI DeepTrack 2, and it's also pip installable with the pip install DeepTrack. And all the material is available on GitHub, is fully documented. We are working very actively to improve the documentation and the codes. So it's very much something that anyone who is interested in microscopy can use. It's also open source, obviously. And then later we developed also another package called DeepPlay. And this one is the one that contains all the information and all the tools to do deep learning with, well, essentially it's improving on PyTorch and Lightning in order to provide a set of tools that make building and managing neural networks much more efficient, in particular when it comes to geometric deep learning, which means graph neural networks. We have a unique set of features that it's probably the best that you can get. And Finally, we have been spending a lot of time teaching deep learning because we were asked some years ago to prepare some PhD course originally in ETH for deep learning applied to microscopy. And then we have given this course in many places around the world over the past few years. And finally, all this knowledge, all this that we have about deep learning and all this experience with these courses has come together into this book, Deep Learning Crash Course, that is now being published. It's published by No Starch Press in San Francisco. And the good thing is that the book will come out in the physical form in a couple of months, but it's already available in PDF. If you're interested in the pre-release version, it's the draft form, which is more or less complete. And the good thing is that for people that get it now, there is a big discount of 25%, and the price is still $60 for the printed version, which probably will raise because the publisher wants to raise it a bit, maybe to $70. So just that you know. Okay, with that, we would like to thank you for listening. And of course, we are here available for questions if there are any.
Thank you, Professor Giovanni and Professor Caro. Yeah, at my end, I still have a few questions. And thank you for the work. I'm wondering whether that book is the first version of it, like as if I've seen it somewhere. But maybe we'll see that later. Mine like comes from the paper. You know how the presentation is handled and how the paper is written. Of course, they are totally different. But in the paper, I saw something about, we know this common behavior from graphs, like looking at the local features of the nodes, but also when we have a graph or two graphs, when you have two graphs with sort of like the same generative parameters, but they are behaving differently. And one wonders what could be the causes of such things, of such behavior. For example, I saw something in the paper about a graph or two graphs with the same generative parameters, like behaving in a different way. And if that is something that you can talk about, I would appreciate.
I'm not sure I understand the question. What do you mean by which paper are you talking about?
The graph, the paper that we are discussing, I'm hoping.
Oh, okay, yeah.
The global graph features unveiled by unsupervised geometric deep learning.
Yeah. So you mean the two graphs can have similar generative parameter but different representation?
Yes.
Yeah. Why? But it depends on what does the transformation. So basically, the idea is that, I mean, if you want to extract information in order to parameterize the structure of the graph in some way, you are not getting information about how close it needs to be to something similar. In some sense, you have to impose that or control that.
Okay.
Maybe
I see.
Maybe if you look at this one. Here, the graph is constructed from the localization, from the localizations, right?
Yes.
So the underlying process is the same, but each time you repeat the said experiment, you get a completely different structure.
Yeah, and I think my intuition grew much from here, but I think why I posed the question, like why I didn't ask from here, was I saw two classes, like different classes. I was like, maybe these are different classes of proteins. But in my thinking, I was like, is it like the same picture we are looking at, but behaving in a different way? And it really sparked the connection to that observation I saw.
So, I mean, think about, so you have a group localization in the two cases. But I mean, if you decide to draw the graph, to design the graph based only on local neighbors, it might be that even if the overall structure is different, the local structure is very similar.
Okay, yeah, okay.
So you can have a spot in which, if you focus on the first three neighbors, no, and in a spot, each localization might have three neighbors at a given distance. But also in a ring, you can have the first three neighbors at a given distance. So you're, in some way, you're not getting overall information about the graphs if you only focus on the local features, on the local properties of the graph. This is a very simple example, no? But I mean, I guess it can be generalized. So the same way, you have to keep information about the overall structure and the local structures, and only the mix of, I mean, and you have to mix the two in a proper way in order to represent the overall parameter that describes that system.
Yeah, I think that puts the light onto the question. Thank you. How about, do you? I know you have a question.
Yeah, I have a question. Thank you for the talk, Professor. Could you please elaborate on the structure of AL and SL? Specifically, how does the Gaussian encoder learn to represent both the adjacency and the node features of AL and SL in the Latin space? Or in the sense that what type of information is preserved or emphasized in each component of AL versus SL during this encoding process?
The information that is passed directly is the information about the connectivity.
Okay.
So basically,
Like,
we don't modify the connectivity of the graph. So when Gaudi tries to replicate the same graph, but just replicating the feature of each node and edge, but not the connectivity. And the rest, it goes through this downsampling and upsampling. So the information, I mean, the relational information, it's imposed, but it's not something that the graph learns directly during the machine learns during the autoencoding. Are only the features of each node that are learned. Of course, those features depend on the structure itself, because the way the graph neural network works. So basically, in the update of each node, you get information about the neighborhood. So in that sense, you get information about the relations, but you do not learn directly the relations.
You also talked about the deep track tool that focuses on generating, stimulating, and something like pre-processing of image data pipelines. But how does it specifically support the development and application of the models like the MAGIC for cell tracking, or even the Gaudi for the analysis, for the SMLN analysis?
Yeah.
Especially like when you're considering the challenges of data security mentioned in your take-home messages. So how does it specifically support the development and application of models?
I'm sorry. I mean, I guess I have some connection problem because it gets interrupted. Can you repeat the question?
Okay. I noted down that
yes,
you mentioned that the deep track tool focuses on generating, stimulating, and pre-processing of image data pipelines. Yeah. So I'm asking, how does it specifically support the development and application of some models like MAGIC for cell tracking?
Okay, so the deep track basically it's a way of simulating data. So imagine that you want to, you have whatever problem in microscopy you want to solve, you don't have labeled data, you can simulate realistic data. So basically you can train whatever model, like, whatever model associated to the deep learning in microscopy, having labeled data generated artificially, synthetically. So what deep track does is this. It's basically a tool to generate physics-based images. So basically, you have a feature that are like a microscope with specific optics. You can change from bright field to fluorescence, and you can create objects and apply the diffraction and apply any kind of physical noise. And then you can recreate something that looks like a real image. And you can do that easily, for example, for physical object like beads and or ellipses, but by combining this object, you can create living objects. For example, we have been able to reproduce images of cells, of plankton, or bacteria, and so on. So basically, this deep track is a tool that helps you to get without doing experiment and without manually labeling them a starting training dataset that looks like the data that you want to apply your machine to. And this is deep track. Then for the architecture that we use deep play. Deep play is basically a tool based on a library based on PyTorch that simplifies the process of building a deep learning tool. In the sense that you have it's very modular. You can you have a lot of bricks that are already prepared. You have architecture that are already prepared. And basically, you can train a network with one line of code. For example, if you want to train a unit and you have already the data, you can use one line of code in deep play and train it. So just a way to simplify the use. So those are the two tools that we mentioned. But one is more for generating data, and the other one is more for building architecture. I don't know if this replies to your question.
Yeah, sure.
Thank you, Albert. Prof, I think for me, I have two questions. One, somewhere in the slides you talked about morphology encoding, I think, and I'm wondering if we have two like sort of similar graphs, but with sort of thinking about them in a different morphological structure. If we are to encode morphology, how do we make sure that it is preserved within the training or within the learning process, and we can account for it within the results? Then another question I think is about the ring and spot classifications. Maybe throwing about more light about them, like those two different classifications. Yes, I understood the ring, sorry, the spot, and how it gets tracked within the different sort of frames, but I think I missed the ring part. So if you could explain it more, I would appreciate. Those are my questions.
So regarding morphology, let's suppose that we have some cell that is moving, and we want to track. So basically, we want to connect the cells at different times. And of course, these cells will have some feature that is somehow different from another cell. That can be a morphological feature. So we can just use some pre-processing of the ROI of that cell, get the number out of it or a set of numbers, and put it as a node feature. This is a simple way. You want to complicate it further, you can apply a convolutional neural network to the ROI, get a set of parameters that associate, for example, you can use a ResNet or whatever other classification network. You cut it at some specific layers. You get the feature from that layer, and basically this is a representation of that ROI through a neural network that has learned some feature. Again, you end up with a vector that you can put it as a node feature into Magic. And if the cells changes, but morphology, but does it progressively, the network is gonna learn this change, and it's gonna learn that this feature is modulated over time in some black box way. So this is about morphology. Regarding ring and spot, so basically, in a biological cell, you can have proteins that aggregate in different ways. Typically, the simple way for the protein to aggregate, for example, at the cell membrane, is just to get together and form a spot. But in other cases, like in nuclear pore complexes, they aggregate in spots, but then these spots then form a ring. And this is a function because basically this is a pore. This is something. This is a hole in the external part of the cellular nucleus that let other molecules to pass through or not. And basically, what we want to do in this case is to learn about the ring as a whole and classify different rings when they are very close together. But at the same time, we want to be able to quantify, for example, the number of proteins that form each spot that has this 8-fold symmetry that forms the ring. How do we do that? Basically, we divide the supervised process into steps with two loss functions, one after n iterations and recurrent steps, and the other one at the very end. In this way, the graph neural network progressively starts to shrink first the spots and have this 8-fold symmetry, and then get together the spots into the final cluster. But it's supervised.
Wow, that's great. Thank you very much. I think there is a question in the chat. I hope Professor Giovanni noted it.
It is in the chat. Okay.
Yeah. The applications shown for Miro and Gaudi.
Maybe Giovanni can comment on that.
So, Carlo, do you want to answer?
Oh,
I mean,
yeah.
there are the other examples already in the paper.
Yeah. So basically, in the paper we show a few examples, and those are all from the complex systems field. So we start to apply Gaudi to the Watts-Rogatz graphs. Basically, those are graphs that are formed by points on a ring on a circle that are randomly connected, and they can be randomly connected according to two parameters. And in that case, we see that Gaudi is able to cluster the latent space according to the two parameters that define these graphs. Then we also apply to the diffusion of active matter. Basically, we simulate data according to the Vichek model. So basically, those are like bird flocks that, you know, they have some self-organizing principle, and they start they flock when they fly, because they start flying in the same direction. So they form these dynamic clusters. Also in that case, we simulate the data, and we see that Gaudi is able to cluster in the latent space graphs that correspond to a Vichek simulation with similar parameters. And then there are these two applications that we showed, one of the single molecular organizational microscopy and one of the brain connectivity dataset. But in principle, everything that can be represented as a graph can be
yeah. If you think of a biological application, you can think about networks of proteins, ecosystems, food webs, even human networks, starting from social networks. So anything that is represented by a graph in which you can assume that there is an underlying mechanism that produces the graph and you want to discover it.
Wow. I hope that answers the person who asked. We have shot beyond the 1 hour mark. I want to thank Professor Caro and Professor Giovanni for saving time on their tight schedules and really share with us this interesting knowledge. Thank you very much, everyone who has attended and participated. And great thanks to you, Professor Caro and Professor Giovanni. Thank you very much. Thank you very much indeed.
Thank you very much.
Thank you.
Thank you. Bye-bye.
Bye.
Bye-bye. Yes.